Hello Bruins, and this is going to be a presentation on Mendelian genetics and extensions that don't follow the classic complete dominance. So hopefully after this presentation you'll be able to do dihybrid crosses to analyze pedigrees and know inheritance patterns such as incomplete dominance, polygenic, and sex-linked. Now, Mendel argued that parents pass their offspring some kind of discrete heritable factor, and today we know that heritable factor, as you can see on a picture, is genes which are found on a chromosome. And notice I have a picture of homologous chromosome. So homologous, remember, means they're similar chromosomes with similar information, but they're not identically the same because you can have different versions of the genes. So here, for example, everyone has a pair of homologous chromosome, and we have 23 pairs. So it has genes on these chromosomes, and these genes are represented by letters. And genes are then expressed to a trait or a protein. Now you have different versions of a gene, which are called alleles. Now these alleles can be dominant, right? Or you can have a recessive allele. So in a complete dominance inheritance pattern, if you have the dominant allele, it'll show up, and if you have two recessive alleles, then the recessive trait will show up. And then we have other words to describe our genotype, or what alleles that individual has. So if it's homozygous, it'll be big P, or small a, or if it's heterozygous, it'll have two different alleles. Okay. So if you remember from our discussion, Mendels Law of segregation relates to how those homologous pairs are distributed to gametes. So here it shows you have a purple flower, which is purebred. So you see it's homozygous, purple, and white flower, which is a recessive trait in this case, and it's homozygous. So remember, whenever you have a recessive trait, it's always going to be homozygous. So when you mate the purple flower and white flower, or you cross, you'll have the F1 generation being all hybrids. Now those gametes within this hybrid plants, their alleles are segregated. Remember, during anaphase, the chromosomes segregate, go to opposite poles, and then each cell will get that chromosome. And so if you have many chromosomes, all those chromosomes will segregate independently. So if you have a hybrid and a hybrid cross, you will have a 3-to-1 ratio, 3 that are purple and 1 being white. So that's called a phenotypic ratio. And remember, phenotypic means what it looks like. And here if you see, this is a genotypic ratio. And the genotypic ratio, what are the alleles? Do you have homozygous, heterozygous, or recessive? Okay, and that leads us to the law of independent assortment, which means that each allele that is on a different chromosome is going to be assorted independently. Now, this comes to our discussion, what happens if we look at two characters at once? And so what Mendel thought that, hey, if I cross a round, smooth P with a wrinkled green P, then I should get all gametes combination of big RY and small RY. And all the F1 generation should be hybrid. And so this is the Punnett Square that he was thinking. But it didn't turn out that way. Instead, he got 4 phenotypes. He got yellow round, yellow wrinkled, and green wrinkled. So he had to kind of rethink his mathematics and said, wait a minute, these gametes are being assorted differently or independently on each chromosome. So it's not like this. Instead, you have 16 outcomes, okay? 4 different ways each parent can have their gametes assorted. So let's do it long way here. Okay, so if you have two parents that are homozygous round, so this parent would be big R round, homozygous yellow, and this parent is recessive, right? And all their offspring is going to be hybrids. So the hybrid would be written like this. And if we cross a hybrid with a hybrid, what should you get? So first of all, how do you assort each parent's gametes? So have a system that works for you. So what I like to do is take the first letter and the second letter, so that's big R, big Y. Then the second letter and the first, that's small r, big Y. Now let's do this, the first letter with the second one, so that's big R, small y, and the second with the second, and that would be small r, small y. So there are 4 possible combinations. So let me do this again. So this is one parent. Now this parent, we do the same thing. Take the first with the first, so that's big R, big Y, and then the second with the first, that's small r, small y. Then take the first with the second, that's big R, small y, and then the second with the second, that's small r, small y. So these are the possible combination. Then the next thing is going to bring all these down and across. So this is going to be big R, big Y, and big Y. And it's a good idea to write the capital letter first. So here is big R, bring that down, small r, big Y, big Y. Bring this big R, bring that R across, big Y, small y, big R, small r, big Y, small y. Alright, so you keep doing this. Bring the big R, big Y down, small r's, big Y, small r, big Y, big Y, big R, small r, big Y, small y. Bring all these down, small r, big Y, small y, big R, big Y, small y, big R, small r, oops, big Y, small y, big R, small y, big R, small y, small y. Okay, once you've filled out your Punnett Square, you bring them all down in a cross, and you write down your 4 possible phenotypes. So I always like to start with dominant, yellow, round, green, then dominant, recessive, green, round, and then recessive, dominant, yellow, wrinkled, and then green, wrinkled. So these are your 4 possible phenotypes. And then anytime to be yellow, it has to have at least one big R, right? In order to be round, it has to have at least a big Y. To be green, it has to have 2 small y's, and to be round, it has to have at least one big R. To be yellow wrinkled, it has to have at least one big R, and to be wrinkled, oops, the other way around. To be yellow, one big Y. And to be wrinkled, oh, it has to be 2 small r's. So to be wrinkled, small r, and to be yellow, at least one big Y. And to be green, has to have small y, and wrinkled, small r. Okay, so if you go down, this one will be yellow round, yellow round, yellow round, yellow round, yellow round, wrinkled, yellow, yellow round, yellow wrinkled, yellow round, green round, yellow round, yellow wrinkled, and green round and green wrinkle. So if you count this, how many yellow rounds you have, it should be going to be 9. And how many green rounds, you have 3. And how many yellow wrinkles, there's 3. And green wrinkled will be 1. So whenever you have 2 hybrids crossing, your phenotypic ratio would always have a 9:3 to 3:1 ratio. Okay, so let's practice another dihybrid. So in Mendels pea plants, axial flower position is dominant to terminal, small a. Green pods are dominant to yellow pods. What is the expected phenotypic ratio of an offspring of a cross between a homozygous axial and heterozygous green plant, and a heterozygous axial and a homozygous yellow? Show the genotypes of the parents and the Punnett Square of your work. So genotypes of the parents is basically translating. So homozygous axial means axial is dominant and it's homozygous, so that's going to be big A, big A. Heterozygous green, and green is dominant, and heterozygous means big G, small g. With a heterozygous axial, with the homozygous yellow, so it's small g, small g. So what would be our possible offspring? So basically what you're going to do is first take one parent and write all the gametes, possible gametes, and then the second parent, the possible gametes, and put it into a 16-box Punnett Square and figure out the phenotypic ratio. Okay, so to set up the Punnett Square with these parental genotypes, so first let's figure out the gametes for each parent. Okay, so here we have homozygous axial with heterozygous green. So I'll take the first one with the first one, so that's big A, big G, and then the second A with this. That's again big A, big G, and with the first one with the second one, big A, small g, and again big A, small g. Okay, so that's one parent on one side. Now I'm going to do the same thing with the second parent. First with the first, that's big A, small g, and then first with the first, small a, small g, and now the first with the second, big A, small g, and then the second with the second, which is small a, small g. And then I'm going to bring this all across. So big A, G, and this is big A, and that's big A, big G, and that's big A, small g, big A, small g, small g. So what I'm doing is I'm taking this across and each one down in each column. Okay, so this one will be big A, small a, big G, small g, big, and then small g, oops, all right. And the possible phenotypes, let's see, dominant, so that's axial green, and then dominant recessive, that's axial yellow, and then recessive, that's terminal flower with green dominant, and then two recessive, terminal yellow. Okay, so these are my 4 possible phenotypes. So to have axial green, I should have at least 1 X and 1 G. And to be axial yellow, it has to be at least one big A and GG, and this has to have small a, at least one big G, and this has to have small a, small g, small g. Okay, so let's see what we have here. So our phenotypic ratio, so this is going to be axial green, 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 axial green. So that's 1, 2, 3. That's 1, 2, 3, 4, 5, 6, 7, 8. Okay, axial yellow. That's axial yellow, 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 and that's going to be 8. And we have 0 terminal green and 0 terminal yellow. So our phenotypic ratio is 8 to 8, 0, 0. Okay, now this shows what happens if we have a dominant trait with a Y trait, and this now is looking at monohybrid, looking at just one trait. Now we know that this purple flower has at least one big P, but we don't know if it's homozygous or is it heterozygous. But whenever you have a recessive trait, you always know its genotype because it's going to be homozygous, small p, small p. So what you do is something called a test cross, and you test cross it with the recessive trait. So if I test cross this purple flower with the white, and I know I got small p, for this to be all purple, I would have to have homozygous. So if all my offspring are purple, then I know it's homozygous, big P, big P. But if 50% are purple, that means I have to be heterozygous, and I would have 50-50 ratio, or 1-to-1 ratio, where this would be 100% purple. So this is called a test cross. You can also do test cross with other unknown genotypes. Here, for example, you have a dog, black dog versus brown dog, and we test cross it with a brown dog because that's recessive, and if all the offspring are black, you know he's dominant, and if the offspring is 50-50, then you know he's heterozygous. So when they're trying to get a purebred, this is what they're doing. Now Mendels Law follows the rules of probability. So basically what you're doing is when you're making a Punnett square, you're trying to calculate the outcomes, the possible outcomes of the offspring from the gametes you have. So if you think of a penny, you have half a chance of being heads and half a chance of having tails. Same thing like with the gametes. So here with the dog, if he's heterozygous, I have half a chance of that gamete being a big B. I have a chance of it being a small b. Now whenever you have two independent events, you multiply, and the product is the possibility for each event. So here half times half gives you 1/4 to be big B, big B. 1/2 times 1/2, 1/4 chance, and so forth. Now what happens if the probability can be a either or, like it can happen this way or you can have it this way. In this case, to be heterozygous, you will add the 1/4 plus 1/4 and you'll get 1/2. So you have 1/2 the chance of being heterozygous with two heterozygous parents. Okay, now let's move on to pedigrees. Now there's a different way besides making Punnett squares. You can chart out a family tree using a pedigree chart. So we use symbols. Circle and colored in is the affected female. A square colored in is the affected male. A circle female that's not colored in, or a square that's not colored in, that means it does not show that particular trait. Now when you have a line between a square and a circle, that means they have mated, and you bring a line down and these are their offspring. And here you can see you have two couples, and this male has mated with this female to have these two offspring. So this is what's called a pedigree. Now, how do you know if you're looking at multiple generation, do you have dominant or recessive? How can you figure that out? Now, if you see the parents don't have this trait in this case, and you see it coming in the offspring, it must be a recessive trait. And if both parents don't show it and it shows up in the offsprings, they must be heterozygous. Because in order to show a recessive trait, both parents have to have that recessive allele. Okay, so in this case, this parent shows a trait and this parent is heterozygous. Therefore, this individual has the trait. Okay, so in this case, we're looking at attached years versus free years. So attached years is recessive in this case, and free is dominant. And the way we would label recessive dominant is it's on a chromosomes 1 to 22. So we will say this is autosomal recessive trait. Okay, sometimes you don't have a complete dominant trait. So in the case of carnations, here you have a red flower. Now you can use a big R or R prime or RW. So let's use the R and W so we don't confuse between the complete dominants. So if it's big R, the two big red alleles, then it's a red flower. And if you have WW, then it's a white flower. Now what happens if you're heterozygous and you have one R and one W? It's an in-between trait. They have this blending effect. So it'll be pink. So notice it's not a complete dominant, but an incomplete dominance. So basically what we did, we have a pure red flower, big R, W, W. And all the flowers are hybrid. And if you cross it with another pink flower, then you would have a red flower, two pinks, and a white. So your phenotypic ratio, big R, RW, WW, red, pink, and WW is white. So here you have phenotypic ratio and your genotypic ratio. So this is your genotype, and this is your phenotype are the same. The phenotypic and genotypic ratio are the same. So when you see a third phenotype, that'll indicate that it could be incomplete dominance. And if you see that blending effect, an in-between trait, then you know it's an incomplete dominance. Now what if that trait is a codominant? So that means that if you have that gene, you show it. Alright, so there's no, both traits show up. Now, blood is an example of codominance and also multiple alleles, meaning that you have multiple genes that will indicate your blood type. Now, what does blood type mean? If you're blood type A, and usually we show our alleles AA, or you could be A or O when you have no subgroups. So what happens is A means you have a particular carbohydrate present on the red blood cell. And if you get in a B blood, that will produce antibodies against the B blood, and it starts coagulating. It won't react to O, and it won't form any antibodies against A. But any B will form antibodies. Now if you have B blood, you will form an antibody against A. And AB has both carbohydrates A and B, and it will produce no antibodies. Now if you have O blood, you have no carbohydrates, but you will form antibodies against A and antibodies against B. So this is the test they would probably give you to figure out what blood type you are. So for example, if you know, say your mother is blood type A and the father is, say, blood type O, what are the possible blood types of the children? Here's my Punnett Square. Now the mother could be AA or AO, and the father, of course, is OO. So in this case, the offspring can be heterozygous, it'll be AO, or they could be OO if the mother is heterozygous. So the possible blood types for the offspring is AO, OO, that's their genotype, and phenotype is either A or O. Now, what happens when you have traits that are linked to a chromosome? So for example, sex-linked, so remember chromosomes 1 to 22 are autosomes, and chromosome pair number 23 is a sex chromosome. So if you're a female, you have 2 X's, and if you're a male, you are XY. Most of the sex-linked disorders are X-linked, meaning they're on the X chromosome. So for example, colorblindness. Can you read the letters on this slide? Now if you can, you're not colorblind, but if you cannot, then the likelihood that you are colorblind. So you should see a 12 here and a 29. Now how do you express sex-linked traits? How would you write a Punnett square? So here, for example, let's say if you have a colorblind male, and we would write the sex chromosome, and we're gonna write, I'm gonna use B. So big B is normal, and I'm gonna write small b would be colorblind. So the possible phenotypes that I can have is female, normal, female, colorblind, male, normal, male, colorblind. Okay, now in order for a female to be normal, since she has 2 X chromosomes, one of the X chromosomes must have a normal gene, and the other one, so she could be homozygous normal, or she could be a carrier. For her to be colorblind, she would have to have both colorblind genes. Male, has to have that just one colorblind gene, or normal. Okay. So for this male, if he's colorblind, he would have that colorblind gene, and let's say this female is homozygous normal. So let's write the phenotypes of the female here, XB, XB, and the male would have 1 XB chromosome and Y. So notice the male would never give his colorblindness to his sons. He only gives a Y chromosome. But that X comes from the mother. So males get their X from their mother, or whatever is on that X chromosome. So in this case, the males are normal, and the mothers, the daughters I mean, are going to be carriers. Okay, so the phenotypic ratio, let's put letter A for this. The phenotypic ratio for A is 2 normal females, 0 colorblind, and 0 normal, 2 normals and 0 colorblind males. Now what happens if the female is a carrier and the father is normal? In this case, we'll set up our Punnett square like this. So you have normal female, normal male. Now you have a female that's a carrier and a colorblind male. So this time our phenotypic ratio is 2 normal females, 0 colorblind females, 1 colorblind male, and 1 normal colorblind. Okay, so this is B. Okay, let's say if the mother is a carrier and the male is colorblind. Then in this case, you'll have a a daughter again, that is heterozygous, a normal male. You'll have a colorblind female because she gets 1 from the father, 1 from the mother, and a colorblind male. So in this case, you'll have 1 normal female, 1 colorblind female, 1 normal male, and 1 colorblind male. So you'll have a 1 to 1 to 1 ratio. An example of another sex-linked disorder is hemophilia. Now, a lot of back in those days, the nobility would marry within the family. So here it shows Albert and Victoria are basically cousins. So when cousins marry cousins, you're kind of, any trait that's present will intensify. So the likelihood of, say, hemophilia showing up is greater. So hemophilia is a sex link. It's on the X chromosome and it's recessive. So this daughter is a carrier because she gets it from her mom. And notice the son gets that hemophilic gene, so he has hemophilia. Now, this couple, when they have children, one of their, again, daughters marries a cousin. And then you see, what you can see overall in this pedigree is that mostly who are affected are male. So notice you see more of those squares are affected. Now the female can only get hemophilia if the male has it and if the female is a carrier. You don't see many females having it because if the fathers have hemophilia, usually they don't live long to be a father. Now, other types of inheritance pattern is called, here as an example, is polygenic. The polygenic have an additive effect, meaning there are several sets of genes that affect the phenotype. So here, for example, you have skin color. Now, skin color is affected by 3 sets of genes. So if you're very light, you're all recessive. If you're very dark, you have all the dominants. So the more dominant trait you have, the more darker you're going to have, or the less dominant traits you have, the lighter you're going to be. So what if you take a hybrid, 2 hybrid parents, what are the different phenotypes? So as you can see, you have like a bell curve. So things that are polygenic are like height, weight, skin color, eye color. These are some examples of polygenic genes in humans. Other autosomal disorders in humans, we have disorders that are recessive. For example, albinism, where they lack pigment in the skin. Cystic fibrosis affects producing, you're not able to break down that mucus. That affects your lungs, the digestive tract. Galactosemia, again, you cannot break down galactose, so galactose accumulates. PKU, you cannot break down the amino acid phenylalanine. So a lot of you, when you're born, you get this test to see if you're PKU because you can easily avoid phenylalanine in your diet and avoid having those symptoms. And sickle cell anemia, where your red blood cells sickle due to a mutation. And then Tay-Sachs also is a disorder that cannot break down lipids. So all these are recessive disorders, autosomal. Now dominant disorders, if you have that trait, you have the disease. So Huntington's, Alzheimer's, hypercholesterolemia, okay, and dwarfism. Now also environments can affect the expression of the gene. So here's an example where you have plants, hydrangea blooms. Now they're blue if it's acidic and pink when it's basic. They also do a lot of twin studies to see the effect of the environment. So if one twin does not eat properly, gets exposed to mutagens or smokes, they'll age differently. So there's another example. So this ends my discussion about extensions on Mendelian genetics. Thank you for listening, and please come to class with questions.